Good thing about cell-free therapies is that, as I told you before, you don't need cells. Yeah, actually, that was the same mentor. So this mentor is Professor Gianluca Badala, and
Okay.
actually, he's a really successful orthopedic spine surgeon and researcher in, I would say, in Italy, but also in Europe. And he's one of the key opinion leader in stem cell research for disc regeneration.
Okay, viewers, we are now into another interesting episode in our podcast series. So today we have the guest of honor, Dr. Luca Ambrosio from the Campus Bio-Medico University in Roma, and he is going to give us or elaborate us, or he is going to just iterate his entire journey to success in this next hour to us. So welcome, Dr. Luca, to this podcast, and thank you so much for accepting our invite and being here for us.
Thank you very much, Satish. I mean, I've been really honored by this invitation and really, happy about this nice project that you're running, guys. So happy to contribute to that.
Thank you so much. Thank you so much. So we will kickstart our session. Okay. So we'll be firing you some questions, and we expect your answers as you want it to be. So, like, what inspired you in the first to become an orthopedic spine surgeon?
So, when I was like first, second year medical student, I was actually really into neuroscience. So,
Mm-hmm.
I read a lot the Kandel book about neuroscience, but
Uh-huh.
I also wanted to be a surgeon. So my first idea was, why shouldn't I go into neurosurgery? But then I met, by chance when I was a third year medical student, my mentor who said, one really interesting part of both neurosurgery and orthopedic surgery is the spine, and I am an orthopedic spine surgeon. So why don't you just hang out with me? And that's how everything started. So yeah, I really enjoyed. I mean, I liked the spine a lot, and this was my first interest. And then I had the chance, to kind of understand what was the rest of neurosurgery and the rest of orthopedic surgery, and I enjoyed a lot orthopedics apart from the spine itself, and that's why I got into orthopedics and into orthopedic spine surgery.
Okay, that's interesting. So from third year, you are into orthopedic spine now.
Yeah.
Okay. So, like, is there any defining moment or the mentor that pushed you into research? Orthopedic spine is fine. Into research, how did that happen?
Yeah, actually, that was the same mentor. So this mentor is Professor Gianluca Badala, and
Okay.
actually, he's a really successful orthopedic spine surgeon and researcher in, I would say, in Italy, but also in Europe. And he's one of the key opinion leader in stem cell research for disc regeneration. And, I was engaged, as I told you, when I was a third year medical school. And, in Italy, mostly, I would say the first 2-3 years of medical school are mainly basic science. So when I, started attending the department, I really didn't think I had skills, clinical skills to understand that part of orthopedic surgery, and I really enjoyed basic science because I liked biology, biochemistry, physics. And he was starting at that time our regenerative orthopedics lab. So I really asked, instead of starting attending the outpatient, the OR, which I could not appreciate at that time, I started going to the lab and learning the basics of cell culture, gene expression assays, Western blot, and stuff like that. And I really enjoyed that, and that's how I got into basic science. And then as I grew up and had some more experience, I was also involved into the clinical research that we were running, because we are probably the main institution in Europe running the highest number of RCTs investigating disc regeneration with stem cells. So this really, involvement gave me a lot of insights into clinical research, and that's how I got into that field as well. So
Okay.
I owe my mentor everything, basically. In that sense.
So Gianluca has all the credit of
Yeah. He does. He
orthopedic
does.
spine surgeon and the researcher.
Yeah.
Okay, that's great. Okay, then how did you balance to get into the clinical training as well as the research? How did that go?
I would say I still couldn't find a balance, because both are so demanding in
Yes.
terms of time and mental effort, devotion. So sometimes I really feel overwhelmed by all the deadlines and all the duties. So I try to allocate as much time as needed to research. Good thing is that we have a strong group here, so we can also, let's say, split the duties among us, and this is key, I think, because you cannot do that by yourself. You need to you need a team. You need to educate students to help you. So I would say there are some periods which are more balanced, some others like this one now, which is totally crazy with deadlines and stuff and project proposals, grant submissions, and you just have to cut time, unfortunately, to your personal things. So you need also, I know, a lot of support from your family, from your friends, and it's a system that you really need to build in order to make it work. It doesn't work everywhere every time, because it's everyone is asking more from you. But I mean, that the main challenge is to try to balance between your personal life and research, and of course clinical stuff, because patients are always our first priority. So I would say balance is not a thing that I really have now, but I strive to get that.
Okay, thank you for a very honest answer.
Yeah.
Because my question is another scale. Like, apart from balancing between the clinical training and research, you need a personal life that also needs a balance. So
Yeah.
it's always difficult, and everyone is trying to materialize that in one way or
Yeah,
the other.
I think that's the main fuel. You cannot get anywhere by yourself. You need
Yeah,
support from your family, from your friends, from your wife or husband, from your kids. You cannot make it basically without motivation from that side.
yeah, yeah.
And you have to be lucky, because it's not up to you.
I totally agree to that. Okay. So the next question is Gianluca Vadala also the reason behind your choice for regenerative medicine and the disc biology research, or is there any other person who is also driving your focus towards that?
So again, I think this started from Gianluca, because he was and is one of the main people in our field in that sense. So when I started, actually participating into the research activities, of course, that was our main focus. And also, what I did later in the US, in Switzerland, and in Japan was always, more or less related to disc regeneration, and that has been my main interest since then. So what I like the most about that is it's a really challenging environment, I mean challenging field. So and the fact that we are really not finding an answers, it's what pushes me to still try to look for that, and that's why I'm into that.
Okay, okay. Thank you. Like, what is the role of your institution, like the Campus Bio-Medico University, is playing a role in your research and clinical activity? Like, how does
Yeah,
it
I
support?
would say, I mean, I have done my medical school here, my residency here, and I'm practicing here. So I was literally born and raised into this university, and that was because I found here a lot of mentors, but also possibilities. So Campus Bio-Medico is not an old university, because it was started in 1993, and it's a let's say now medium size, I would say, private university in Italy with a great devotion to teaching and students. So now it grew bigger since since I left medical school. But one of the main characteristics of this university is that the numerical ratio between the professors and the students is really low. So I mean, there are a lot of professors compared to the students, and this allows our students to be involved and looked after and engaged in a lot of activities. What happens in bigger universities that there are so many students that you maybe end up being just a number. And what I really enjoyed here was that I had so many opportunities that I could take, and that's probably what led me here. So that's why I'm really grateful to my uni for giving me all these possibilities. So I would suggest anyone interested in this university who has the possibility and the will to join, just to do that, because it's a great one.
Okay, thank you. So let's go into your ISSLS Prize winning project. So for those who doesn't know, ISSLS International Society for Studying the Lumbar Spine, and they usually have their annual conference where they give the best project ISSLS prize. And it is this time the Lucas project that was awarded the ISSLS prize. So we just want you to brief us about your project and why you chose this MSC-based EVs and why those cells and what is promising about those cells on the intervertebral regeneration.
Okay, so this started actually from a research fellowship that I won in 2021, I think, from the North American Spine Society. Again, to all people that are listening, this is a really great opportunity. Every year, the NAS has these grants for fellowship and projects, and as an average resident involved into research, I was looking into that. I applied, and I encourage you to do so. And once you apply, you have to propose as a research center you want to visit. And I've always been in love with Japan. And actually, there was this publication from Professor Sakai, which I later visited, which was published in Nature Communications in actually 2012. And he described for the first time the presence of local progenitors with stem cell-like properties within the disc, which were characterized by the expression of the Tie2 receptor. So since we are always been interested here in mesenchymal stromal cells derived from mainly the bone marrow or the adipose tissue, when I read about these, specific progenitors, I was really, interested. And then there have been further studies demonstrating that compared to bone marrow MSCs, these Tie2 positive progenitors have a better chondrogenic capacity and a higher committed phenotype to the nucleus pulposus cell line compared to BMMSCs itself. So I was really interested into that. I love Japan, and that's why I went there. And Professor Sagai is such a visionary person. He accepted my research plan. We started to do in vitro studies, and at that time I was really interested into extracellular vesicles, because the idea of building up cell-free therapy without the hurdles and the costs and the time-consuming steps needed for cell expansion really interested me. And this is also, a great trend into regenerative medicine. So what I proposed was, why don't we look into extracellular vesicles secreted by Tie2 positive progenitors? And of course, we needed a comparison, and this was in our case BMSCs. So we compared EVs from Tie2 cell positive cell progenitors versus EVs from BMMSCs. And we found out that in vitro and also in vivo, Tie2 cell-derived EVs were significantly more effective compared to the BMSC EVs in terms of, in our animal study, preservation of disc structure, disc height, preservation as well. And also what I really was surprised about was that when we were doing behavioral studies, the animals treated with these EVs showed less local pain behaviors compared to the other animals. So that was really nice adventure. And when we closed the in vivo study, we had a lot of interesting data, and Professor Sakai just suggested that we could try to submit that for the ISSLS Prize. And then when we had the news, I was like, oh my God, this is not possible. The ISSLS Prize for us is like the Oscars, no? So that was great. That was the start of, the second chapter, because thanks to that publication, I was able to get another big grant from the NAS, which is now funding our study in a bigger animal model, which will be next year. Which will, bring me back to Japan, and so many things are moving on thanks to that. So I'm really, grateful to the ISSLS and all the, possibilities around that. So again, I really, encourage you to be involved in that society and to send out your papers for being considered for the prize, because, it's like a domino effect. So I really wish that everyone can get that.
Thank you so much for sharing your experience and the opportunities available for the young researchers to pitch their project and get it taken forward. So Having seen the effectiveness of these cell-free therapies, how do you see it evolves in spine surgery in the future? Do you see them to be the promising future therapeutic scope in spine surgeries?
It's really difficult because actually the good thing about cell-free therapies is that, as I told you before, you don't need cells. And when you have cells, you have a lot of time-consuming steps to be undertaken, considering the expansion, the isolation, and all that stuff related to that, and also some anecdotal but real risks regarding immunogenic and tumorigenic concerns. So with cell-free therapy, you actually abate all these drawbacks. But of course, cell-free therapies, they only work if they find cells into the tissue. And we know that with advanced disc degeneration, there are almost no viable cells into the disc. So of course, I see future for this application, but probably only for early disc degeneration, because until there are functional cells that can actually absorb the EVs, that's how and when EVs can function on these cells and can exert their effects. But if there are not any more cells there, EVs, they just float into the extracellular matrix without doing nothing, because they cannot be absorbed by any cells. So what I see in the future, and it's also something I would like to explore more once we finish this project, is really designing an integrated solution, because when we have this degeneration, we don't have cells, we don't have cues from the extracellular matrix, and the extracellular matrix itself is degraded. So what I'm trying to designing in my mind is probably a solution including a biomaterial as a scaffold that can both restore the biomechanical properties of the disc and serve as a scaffold for the cells, functional cells, and in the same prop that EVs. So I was thinking of probably some way to make these three element product injected into the disc, and maybe by means of some magnetic molecules, trying to make cells and EVs into contact, like applying some forces to the locally injected biomaterial, make them into contact, and activate the cells into the biomaterial after injection. So I really think that as researchers, sometimes we are really having a partial view of the problem. Some are working on biomaterials, some others are working on cells, some others on cell-free therapies. We are probably missing the landscape. So I think that is the future.
Okay. So the future of biological therapy for spine surgery is not just the EVs, but it is complete tissue engineering, where you need a scaffold, a cell, and the EVs or whatever may be the cues for it to regenerate the population that is lost due to degeneration. Okay, thank you for that wonderful explanation. So you said that you are a host of a lot of clinical trials on regeneration in your university. Like, we have seen a lot of disc regeneration failures. So can you identify in your experience what is the reason behind that, and what can be done differently, and what are the possible challenges that needs to be met in the clinical trials for regeneration in disc?
Yeah, thank you for the question. So this is a really interesting topic. I'm really into that, and we have recently published also a study in George Spine regarding all the possible reasons why these clinical trials fail. So one first thing is that when we are working in preclinical models, we are dealing with disc degeneration. But when we go to patients, we are including patients affected by low back pain. But disc degeneration is not a synonym of low back pain. As we know from the clinics, many people, they have disc degeneration and no low back pain, but so many people have low back pain and no disc degeneration. But of course, the clinical problem is not disc degeneration itself, it's low back pain. And we don't have the knowledge of this relationship to that point that allows us to include the patients that really deserve a biologic treatment, because a lot of patients have low back pain due to different reasons, and probably having a biologic therapy in their discs is not what is healing the cause of of their low back pain. So I think that the first big challenge is trying to find a way to really select those patients affected by pure discogenic low back pain, if ever possible. Second thing is that the quality of the evidence is not that high, because there are so many small studies, and some of them are industry funded, and we have that when COI is a thing, everything gets a little bit cloudy, and that doesn't contribute to the reputation of regenerative medicine in general, because when these badly designed trials fail, the overall feeling from the field is that regenerative medicine is just not working. So I think that the biggest challenge is patient selection and product optimization, accurate description of all the experimental steps leading to the preparation of the product, also to allow reproducibility and reliability of what's been done. Of course, long follow-up and reasonable outcomes and honest conclusions, because at this point we don't have the evidence to say that these therapies are working on chronic low back pain. So I think that these also, we have a lot of publication bias, because of course negative studies are not being published. So I think that the overall perception of the field should be more honest and more scientifically driven in to encourage further advancements.